Immune-related diseases account for 3 of the top 10 leading deaths in the world. Our immune system has historically been our best defense against new diseases and microbes. Yet as these disease cells continue to adapt to our immune systems, it will become crucial that we find a method to enhance our immune system. One enormous problem is that these certain disease cells or antigens within our body, they will alter the surface of their cells and change their receptors to obscure the binding factor of our white blood cells. This is problematic, since now our white blood cells can't bind to these viruses and microbes, leading us to become susceptible to the effects of these diseases. This is how various forms of cancer work and why it's so hard to deal with cancer and other diseases that have obscuring binding receptors. Chimeric antigen receptor T cells, or CAR T cells for short, are genetically modified white blood cells that can be programmed to bind to certain disease cells within our body. We can alter the receptors of our own body's T cells so that they can bind to these antigen-presenting cells, or APCs, and trigger apoptosis. The reason T-cell therapy hasn't been able to work in the past is because finding which receptor that will bind to a certain APC is difficult and really hard to predict. The purpose of my project is to propose a solution to predicting the specificity of these T-cell receptors given a certain antigen. The mechanism in which to do so is to use machine learning, a subset of AI. Various models were created to predict the various parts of these T-cells. More specifically, the parts of the T-cells that have the biggest effect on the binding factor of the receptor, which include the third complementary determining region, or CDR3 protein sequence, and the variable segment and joining segment, which make up most of the alpha and beta chains of the T-cell receptor. We want to predict these parts given the antigen-presenting cell, or more specifically, the epitope protein sequence, which uniquely identifies and defines an antigen-presenting cell. So given the epitope protein sequence, can we predict the CDR3 protein sequence, variable segment, and joining segment of a T-cell receptor? And to make this task even easier, we can divide our large task into 2 subtasks, creating a model for the CDR3 protein sequence and creating 2 models for the variable and joining segments. The CDR3 sequence has a very different structure compared to the segments, which is why it makes sense to break up the tasks. For the CDR3 protein sequence, a modified machine learning model was implemented. The epitope protein sequence and CDR3 protein sequence were represented by encoding our protein sequences with a large letter-to-number encoder, and then creating an embedder that maps relationships between data together. A modified sequence-to-sequence learning model was implemented that used more LSTMs to figure out which protein sequence would uniquely take away that one antigen. For the variable segment and joining segment, a classification-based model was created because there are only a discrete amount of segments that are applicable to some of these TCRs. The epitope protein sequence is represented in the same way. However, the segments are one-hot encoded in order to create distinct vectors that constitute class. A standard convolutional neural network plus a fully connected neural network was implemented to classify an epitope protein sequence to the corresponding segment. After training all 3 models for 30 epochs each, the CDR3 sequence model was able to reach an accuracy of 87%, and the segment models were able to reach an accuracy of 80%. Here are a few examples of the models in action. Here, it specifies the TCR for the melanoma antigen. Here, it specifies the TCR for the herpes virus antigen. And here, it does the COVID-19 antigen. If you would like to try out the model for yourself, you can download the weights that were provided in the submission, go to this Google Colab link, and load the weights. Then run the cells and input your own epitope protein sequence to figure out which certain TCR will bind to that certain epitope. This project was created to fundamentally change the way we deal with immune-related diseases and allow us to leverage the best weapon of all, our own body. I hope this project can help bridge and solve a major hurdle in the field of T-cell therapy and completely revolutionize the use of this innovative medical procedure. Thank you for watching this video, and I hope that some good change comes out of this project.